In this video, I'm going to illustrate how to do a monohybrid cross using a Punnett squared. So it's really essential that you're organized when you are trying to solve genetics problems. Typically, it's going to be a story problem where you get a lot of information about the parents. And then you're asked to determine the expected genotypic frequencies or phenotypic frequencies of their offspring. So I've got this problem set up, and this is kind of like one of the monohybrid crosses that Gregor Mendel did. So he was crossing pea plants, and he always used true breeding parents. So I've used P0 to designate the parental generation. So his true breeding purple flowered parents only made offspring with purple flowers when they bred with themselves. And his true breeding white flowered parents only made white flowered individuals when they were bred with each other. And that's because these individuals were homozygous dominant in the case of the purple colored plants, or homozygous recessive in the case of the white flowering plants. So they didn't have any genetic diversity. So when they bred with each other, they were true breeding. They always, in the case of the purple flowered individuals, had offspring with purple flowers, and in the case of the white flowered individuals, had offspring with white flowers. So in genetics, you can see here from looking at the parental generation that we always use capital letters to denote a dominant trait, and we always use lowercase letters to denote a recessive trait. Mendel knew that purple flower color was dominant and white was recessive because he crossed these two true breeding parents, and he found that their progeny, or the F1 generation, was all purple. So their phenotype was they made purple flowers. Their genotype was big C, little c. So they had inherited one of the dominant alleles from one parent and one of the recessive alleles from their white flowering parent. But because of the law of dominance, which you read about, in this case, all of these individuals are going to have purple flowers because this dominant allele is masking the expression of the recessive one. So they all look the same. They all have purple flowers. Mendel took this to the next level, and he then crossed the individuals from his F1 generation who are heterozygous for the trait. That term means that they have two different alleles or two different versions of this gene. So the F1 cross is written here, big C, little c crossed with big C, little c. So in order to determine the F2 progeny, so these are sort of like the parents that made the F2 generation, but in order to determine the ratios of phenotypes or appearances that we expect to see in their progeny, the F2 generation, we should make a Punnett square. And when you make a Punnett square, it's useful, I think, to think about the gamete production of these parents. So I'm drawing little eggs across the top and little sperm down the side. So if we think about gamete production in an individual who has two copies of two different alleles for this trait, their gametes are either going to contain the big C allele or the little c allele. The same is going to be true for the other parent. The female, in this case, is going to make eggs that either have the big C allele or the little c allele. Because remember, the whole point of gamete production is to make haploid cells that just have one chromosome from each set, from each homologous pair. So these two alleles are on separate chromosomes, homologous chromosomes, and in gamete production, they're going to get separated from each other. And that's actually the basis for Mendel's law of segregation. Okay, So now in fertilization, we know that sperm is meeting egg. So we're going to imagine when we fill in the square, we're imagining that this sperm fuses with this egg. They both had big C's. So in this box, I'm going to draw two big C's. Then we're imagining a different possible fertilization event where this sperm that has a big C in it fuses with the egg that has a little c in it. And we're going to go all the way through the square, imagining all the different ways fertilization could possibly happen when we have two parents with this heterozygous genotype. So we've populated the square, and now what we're looking at in the center is all the possible genotypes of the F2 generation. These are diploid individuals. That's why we have two copies of each letter, because they have one copy on each of their chromosomes, on each of their homologous chromosomes. So the last thing we do when we're doing one of these genetics problems is we write the ratio of phenotypes. And I don't think I mentioned this, but it's always really important to stay organized and write yourself a little key so that you know what the combinations of genotypes or alleles that are going to get you each of the phenotypes. So we just need one big C in order to have a purple flowered individual. So I see 1, 2, 3 out of 4 of these offspring being purple. So 3 purple, and then I only see 1 individual in this square that has white flowers. So we would expect for out of every 4 offspring produced in this cross, we would expect 3 to have purple flowers and 1 to have white flowers. And this is the ratio that Gregor Mendel consistently found for phenotypes in his monohybrid crosses for many different traits in peas. So this is an example of how you would use a Punnett square to solve a monohybrid cross.